Hello students, welcome back to my channel. In this video, I am going to give you a brief lecture on DNA replication. DNA is nothing but deoxyribonucleic acid, which is a macrobiomolecule that consists genetic information of a species over millions of years. Okay, so that's why DNA is considered as reserve bank for genetic information. So it is also considered as a memory bank. Okay, so let's see what is replication. Replication is a process by which the DNA molecule can produce its identical copies. Okay, so in replication, one parental DNA molecule that can produce two identical copies of DNA. So that is nothing but DNA replication. Let's see why should DNA replication occurs. There are two reasons. So let's see the first reason. To pass the genetic information to the new cells. So as a part of growth, cells are dividing continuously. So the information, the genetic information present in DNA that should get multiplied before the cell division, because one cell is going to produce two cells in the mitosis. Okay, so the DNA present in the single cell that should be shared equally to the two cells. So that's why before cell division it should get multiplied. So that's why replication occurs. Let's see the second reason, for reproduction purpose. So during the reproduction, gametes are produced as a result of meiosis. Okay, so male gametes and female gametes, they are produced as a result of meiosis only. Here also cells are getting DNA from parental cells. Okay, so when the gametes unite to form a zygote. So now the zygote has the genetic information from both the parents. Okay, so the gametic DNA is produced as a result of replication only. Okay, during the gamete formation also DNA should get multiplied before the gametic formation. So that gametic DNA unite to form zygote. That zygote receives the DNA from two parents. Okay, that's why the offspring having genetic information from both the parents. Okay, so let's see when should DNA replication occurs. As I told you earlier, the DNA present in each cell should get multiplied before the cell division. Before cell division is nothing but interphase. So you know that cell cycle having two phases. Let's see. So this is the division phase, nothing but mitotic or meiotic phase. So this is the second phase, nothing but interphase. So this is the long lasting phase. Okay. So for our convenience, interphase is again divided into three types. So that is G1 phase, S phase and G2 phase. So cell cycle consists two phases. They are interphase and division phase. Interphase again G1 phase, S phase, G2 phase. So G1 phase is a gap phase between M and S phase. So whereas G2 phase is a gap between S and M phase. Okay. So they occur continuously. Whereas S is nothing but synthesis phase. Synthesis of what? Synthesis of DNA through a process called replication. Okay, so the replication occurs during the S phase of interphase. Okay, so you have to remember this. So after interphase only, cell undergoes division, then multiplied DNA. So the DNA multiplied in the S phase, that is shared to the daughter cells equally. Okay, the multiplied DNA that is shared equally to the two daughter cells. Okay, so let's see the detailed mechanism of replication in prokaryotes. So replication mechanism is broadly divided into three steps. There are chain initiation, chain elongation, and chain termination. So let's see one by one. So the first step is chain initiation. So this is the DNA, double helical DNA. So on DNA there are specific points. If you take bacterial or viral DNA, there are specific points. So there the replication starts. Those specific points are called as ori or origin region. Origin or ori. Okay. So at that point the replication starts in the prokaryotes. So prokaryotic DNA consists unique points. Unique point, that point is called as origin. Okay, at this point the two enzymes will come and bind. Those are nothing but helicase and gyrase enzymes. Okay, two enzymes such as helicase and gyrase, they bind at origin point. So they initiates the unwinding of DNA duplex. So when they sit on the origin point, the two strands of DNA, they get separated with the help of two enzymes, nothing but helicase and gyrase. So let's see the separation of two strands. Okay, so this is gyrase enzyme and this is the helicase enzyme. They separates the two strands of DNA. You know, so the helicase that breaks down the hydrogen bonds present between nitrogen bases, whereas gyrase that removes supercoiling. So when the helicase separating the two strands, there are chances for extra twisting of the DNA duplex. That extra twisting is nothing but supercoiling. So that was removed by an enzyme gyrase. So gyrase is a kind of enzyme topoisomerase 2. Okay, it's a type of topoisomerase 2 enzyme. Okay, so it removes the supercoiling. When the supercoiling or extra twisting occurs, the gyrase will cut the DNA and reseals the DNA. So in this way, it releases the tension created in the DNA. So the two strands of DNA get separated with the help of an enzyme helicase. So this separation of two strands is nothing but melting. The separation of two strands is called as melting. So the melting of two strands that creates a replication fork. So this is called a replication fork. A Y-shaped, the melting establishes Y-shaped replication fork. right after the helicase unwinds the two strands. So single-stranded binding proteins will come and bind to the two single strands of DNA. Two separated strands of DNA. So the single-stranded binding proteins, they bind to each strand separately. So why? Because they stabilizes the single strands. So after the separation of two strands, single strand binding proteins will come and they bind to the two DNA strands. They prevents the re-annealing of DNA molecule. That means they prevents the DNA duplex formation. Okay. As a result of the separation of two strands, the bases are exposed to the enzymatic activity. Okay, the bases are exposed to the enzymatic activity. Now an enzyme will come, that is nothing but RNA polymerase. This is also called as primase. So this will come and do transcription. Okay, here it will attaches to the strand. So there it synthesizes 10 to 60 nucleotide RNA strand. This RNA strand is called as primer. Okay, so this is called primer. So when the bases are exposed to the enzymatic activity, in the first, the RNA polymerase will come and it synthesizes RNA transcript, that is nothing but RNA primer, which is having 10 to 60 nucleotide length. Okay, so this gives initiation for the synthesis of DNA strand. Okay, so primer is a small fragment of RNA which is complementary to the template DNA strand, which is synthesized by an enzyme activity, that is nothing but RNA polymerase. So now it initiates the synthesis of DNA. Okay, here you have to remember one thing. The primer is having free 3' OH group. So let's see the second step, that is elongation. In elongation, the nucleotides present in the cytoplasm will come and join to the 3' OH group of the primer. So now the 3' OH group that facilitates the adding of nucleotides one by one to the growing chain. Now the nucleotides will come and join to 3' OH group. Now this nucleotide is having 3' OH group free. Okay, again another nucleotide will come and add to this group. There are 4 types of nucleotides. They were present in prokaryotic cytoplasm. So they are called as deoxyribonucleoside triphosphates. So they are DCTP, DGTP, DATP, DTTP. These are the 4 nucleotides. They are called as deoxyribonucleoside triphosphates. They will come and join to the growing chain. So this leads to the elongation of primer nucleotides in the 5' to 3' direction. You have to remember this direction. Okay. So the direction of DNA replication is always 5' to 3' direction. Okay. So in this way the primer strand get elongated by the adding of nucleotides one by one. Okay. The adding of nucleotides to the primer strand is mediated by an enzyme DNA polymerase 3. So the adding of the elongation step is the elongation is carried out by an enzyme called DNA polymerase 3. So the elongation required free 3' OH group of previous nucleotide. Okay, then only DNA polymerase will add one nucleotide to the growing chain. Okay, DNA polymerase 3 progressively adding nucleotides to the growing chain. In this way, the chain will elongated in the direction of 5' to 3' direction. Okay. So this is called leading strand. The leading strand synthesis occurs continuously towards the replication fork. Okay, it occurs continuously towards the replication fork. You should remember this. Whereas the second strand synthesis occurs somewhat later. Okay, so it occurs discontinuously. The synthesis of second strand occurs discontinuously. So that's why this is called as lagging strand. So the lagging strand synthesis occurs away from the replication fork. So let's see the synthesis of lagging strand. In the lagging strand also, first primer synthesis occurs with the help of an enzyme RNA polymerase. It synthesizes a small polynucleotide chain. After that, so it is discontinuous. That's why another primer will come and synthesizes another primer. It continues it continues up to some distance, and then again another primer will come, another primer synthesis occurs again. It continues up to some extent only. So the action of lagging strand also occurs in the 5' to 3' direction only. So lagging strand generates small polynucleotide chains. Okay, so this is polynucleotide fragments. So these fragments are called as Okazaki fragments. The synthesis of lagging strand occurs somewhat later to the synthesis of leading strand. So the synthesis of lagging strand occurs discontinuously. So that's why it gives us small fragments. Those fragments are called as Okazaki fragments. Okay. So this is called leading strand. It shows the continuous replication, whereas lagging strand shows discontinuous replication. Okay. So that's why the DNA replication is called as semi-discontinuous type. So the semi-discontinuous type replication was proposed by a scientist, Rishi Okazaki. He discovered that one strand of DNA is synthesized continuously and the other strand synthesized discontinuously. That gives Okazaki fragments. So the leading strand moves towards the replication fork, whereas the lagging strand, it moves away from the replication fork. Okay. Later, the Okazaki fragments, they covalently joined together with the help of an enzyme ligase. So let's see the third step, that is nothing but termination. So this is the replication bubble, students. Okay. So it grows in this way, it moves in this way. Okay. So the leading strand moves this side and lagging strand moves this side. Okay. So let's see the termination. So termination occurs when the termination signal will come. Okay. There are some regions, they are nothing but elements, nothing but termination elements. They are localized in the DNA itself. When they reach the termination signal, there one protein will works, that is nothing but TUS protein. So that stops the helicase activity. Helicase unwinds the DNA. Helicase activity is stopped by the TUS protein which is present at a termination region. Okay. So after reaching the termination signal, the primers which were removed by an enzyme DNA polymerase 1. The primers were removed by DNA polymerase 1. So it shows exonuclease activity. With the help of exonuclease activity of DNA polymerase 1, the primers are first removed from the daughter strands. Then the gaps established between the fragments. So these gaps were filled by an enzyme. These gaps were sealed by an enzyme called ligase. Here also primer is removed and the gap will filled by an enzyme. The gap will filled by an enzyme called ligase. So ligase will also catalyzes the winding of two strands of DNA. Prokaryotic ligase requires NAD plus as a cofactor, whereas eukaryotic ligase requires ATP for its activity. Okay. Here you have to remember one thing, that is DNA replication is a type of semi-conservative type. Semi-conservative. You have to remember this. So the Watson and Crick model also suggesting that the DNA replication is a semi-conservative type. Semi-conservative is nothing but half of the DNA coming from the parental strand. Okay, half of the DNA is newly synthesized. So among the two strands, so this is the newly synthesized DNA. Okay, among the two strands of newly synthesized DNA, one is the parental strand and one is the newly synthesized strand. So here each parental strand is conserved and also it acts as a template for the synthesis of new strand. That's why it is called as semi-conservative type of replication. This is about replication, students. If you like this video, please subscribe my channel. Thank you.